Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 107th seminar, and I call it UC Berkeley Day. This seminar is the last one this year, and we'll have 1 month long break. Also, I am so delighted to announce that the inaugural SynBYSS conference will be on December 9 to December 12, 2024 at the University of Hawaii at Manoa. So you are welcome to submit abstract later once that website is open, and then all are invited to join Hawaii conference. Why not? In December, of course, California people not that much into Hawaii conference in December, but many people in cold weather area, and that would be the great escape from the cold weather. I am in San Diego, in California, attending the EBRC road mapping workshop to discuss the vision for space synthetic biology. It's the 8th day of my seminar tour to give 4 talks, all in California, including UC Berkeley, Stanford, Joint Bioenergy Institute, and J. Craig Venter Institute. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Dave Savis. He is a professor at UC Berkeley and also a Howard Hughes Medical Institute investigator, a huge honor for biological researchers. I have known him more than a decade since we met as a graduate student or postdoc. I remember that we had a very nice Korean food together back then. And we talk about, before this seminar, we're talking about Korean food, how amazing that is. So anyway, Dave, thank you so much for your great contribution to the society, scientific society and community, and the virtual podium is all yours now. Thank you.
Thank you, Taesuk, for the generous introduction, and good morning, good evening, everyone. Let me just share my slides here. All right, can you see that? Can you see that okay?
It's coming up. I still see your note somehow.
Strange. Let me try that one more time.
Yeah.
I think it's from the... time.
There we go.
Got it? Okay, great.
Yep.
Again, thank you, Taesuk, for the invitation to give you a short introduction to what my lab works on. And so in the Savage Lab, we think about mechanisms and engineering of CO2 fixation. So probably as many of you are aware, there's a critical, important process of carbon fixation that really powers much of human society. And that is the assimilation of CO2 in its reduction process by photosynthesis, and notably by the enzyme Rubisco, which is the carboxylase driving this process, to produce the reduced carbon molecules that really is the basis of all of the organic life that we see on the planet. And of course, this is a critical biochemical process because we might imagine in the future, even beyond the roles right now, that improvements to this process could ultimately be used to improve the way we produce our food, to reduce the land burden by the intensive agriculture, as well as a tool for CO2 removal. So work in my lab thinks about ways that we can develop basic science and mechanistic understandings of the machinery of this process, and also the tools that might one day be required to bring some of these more synthetic biology type applications to bear in a plant. And principally over the last decade, actually, Taysuk, when we first met, I was probably, I was working on this, and we probably talked a little bit about it when you were in Kristala's lab. My group has been focused on a pathway known as the CO2 concentrating mechanism, or CCM, a pathway that in many microbes enhances their carbon assimilation, such as the cyanobacterium, that are one of the most important photosynthetic organisms on the planet. And the CCM is an interesting physiological pathway that basically couples together carbon transporters, inorganic carbon transporters, with a protein-based organelle known as the carboxysome, a huge macromolecular assembly that contains rubisco. And with this coupling with this transport, improves the carbon fixation process, making it faster and more efficient. So for the last 10 years, we've really been focused on how this process works. We have, for example, carried out high throughput screens in non-model bacteria to reveal all the different genes required for this process, discovering new things like an unknown carbon transporter that is critical for this process. We've also done a tremendous amount of mechanistic biochemistry, for example, trying to reconstitute the carboxysome using purified protein components to understand the interactions and the entire assembly process of this massive organelle that ends up being about 100 times the size of a ribosome. So a massive biochemical system. We've also, as I said, discovered a transporter, and we're working on understanding how this transporter actually works. It's a unique membrane protein with some very cool biochemistry behind it. We're working on that. And finally, with this knowledge, really, as a synthetic biologist, really testing hypotheses to the extreme, we've taken all the genes and all the activities that we've identified, put them all in a system, and reconstituted the entire process in E. coli, actually engineering an E. coli, ultimately, that's capable of growing by fixing CO2 out of the air, as shown here in a recent paper in a collaboration with Ron Malot's lab at the Weizmann Institute. So moving forward, what we want to do is actually take some of the molecules, or at least the principles that we've developed over the years, and start to apply them into plants. As one example of that, highlight the work of Noam Privis, a current postdoc in the lab, who has a paper that he's just posted on bioRxiv that describes the first deep mutational scan of rubisco. These deep mutational scan experiments basically test all possible mutations of an enzyme. And what Noam has found for the first time, as we've systematically tested all possible mutants of rubisco, that of course, as you might expect, there are conserved regions that are extremely recalcitrant to mutations, but also really fascinating positions that will enable us to perhaps evolve the enzyme in a more directed fashion using machine learning tools for enhanced catalytic properties, say improved K-cats, or perhaps more optimal Kms, more higher affinity for the substrate CO2. With this understanding, as well as ongoing work in our lab developing better CRISPR tools to be used in a plant, we hope to combine these two systems to start making edits in plants informed by our biochemistry to create new plants that will have enhanced photosynthesis or perhaps adapted to current climate conditions. And one additional system that we're working on to supplement this is that you all probably understand that working with plants is very challenging. So we're also trying to develop better single cell systems for high throughput screening in plants, such that we can not go through the 1 or 2 year process of making a plant and testing a hypothesis, but ultimately being able to screen millions of cells in order to more quickly understand what types of edits we might want to make before we go through the onerous process of the plant biological aspects. So hoping to bring faster tools to the SynBio community. So with that, I will end and hope you all got a good snapshot of what's going on in the Savage Lab. It's a pleasure to be here, Tae-suk, and thank you for the opportunity.
Thank you. Thank you. Absolutely important and great talk. And also, as you can imagine, given the climate crisis we currently facing, this work is so important, and I hope you succeed, quicker way in near future. In that way, we understand this system better and improve the photosynthesis better. In that way, address many global problems, including climate crisis and also crop yield increase, etc. So that's absolutely important. Thank you so much for your contribution again. Thank you.
Thank you.
Okay, so now the main speaker of today with a longer introduction. Professor Sun Yim is an assistant professor at Korean Advanced Institute of Science and Technology, known as KAIST. He received his bachelor and PhD degree at KAIST. During his doctoral research, he developed genetic parts and tools in Corynebacterium glutamicum and engineered the industrial microbe towards consolidated bioprocessing of raw plant biomass. He joined Harris Wang's lab at Columbia University for postdoctoral training. With Harris, he used a genetic part prototyping approach called DRAFT to characterize thousands of regulatory elements in a single-cell free reaction to rapidly expand the synthetic biology toolbox across diverse bacterial species. He also developed a DNA-based molecular recording framework using the CRISPR adaptation mechanism to store temporal biological information or digital data in the genomes of a living bacterial cell population. Actually, that is one of my favorite work from his lab, Harris Wang lab. He started his lab at KAIST in 2022. Congrats, Sang-sun, again, and has been developing fundamental technologies for microbial systems and synthetic biology to enable scalable characterization and engineering of synthetic and natural microbial ecosystems. Sang-sun, thank you so much for joining us, especially at 1 a.m. in the morning, and please take it away. Thank you.
Right. Thank you so much for the kind introduction. Let me open up my slides here. Right.
Looking good. Thank you.
All right. Hi, everyone. My name is Sun Yim, and it is so nice to meet you all. I'm very grateful to have this opportunity to share my recent work at SynBYSS. It's been only about a year, as introduced by Professor Moon, since I opened my lab. So today I would like to introduce my work during postdoc in the lab of Professor Harris Wang at Columbia University with the title DNA-based temporal recording in living cells. So I'll begin by stating the fact that everyone knows already that the biological processes in native microbial environments are often complex and dynamic. So for example, even a single bacterial cell is densely packed with a variety of biomolecules like this. And when they grow in a biofilm, they do not grow constantly. They rather grow periodically like this to expand the biofilm while sustaining interior cells that do not have access to outside nutrients. And furthermore, natural microbial environments, such as the oral microbiome here, are usually populated by hundreds of different bacterial species that are spatially organized interacting with each other and also with their hosts all the time continuously. So my lab's research interest is in understanding what is happening in these complex environments. And I'm also hoping to take control of the composition and function of these microbial communities for various applications. So to understand these complex environments first, I would like to stress that we need to acquire many different types of biological data. But current approaches to measure these biological data suffer from 3 key limitations. First, many interesting environments, such as the gut, are very difficult to access for direct measurements. And second, most methods require disruptive processing steps like cellular fixation or lysis, and they cannot yield temporal biological data. And third, even methods that allow continuous data acquisition, such as live cell imaging or cellular biosensors, they require direct access to the biological samples, and also instruments to measure these are usually huge, and they cannot be miniaturized down to cellular sizes. So we need some invasive ways to get access to the samples again. And I believe these limitations are preventing us from better understanding about how complex microbiome processes develop over time. And as a microbial engineer, I was motivated by these limitations to explore a new way to measure biological information by measuring things from the inside. So what I want to propose today is this DNA-based cellular recording concept, where resident microbes in the target environments could be engineered with a synthetic DNA-based memory capability to offer the capacity to measure and report biological signals over time. And as you can see in this figure, engineered cells could be utilized for non-invasive temporal recording of important signals such as nutrient status and also microbial or host-derived metabolites. And in addition, these cells could be engineered to detect even some disease states and actuate some therapeutic responses to treat diseases in real time in their native environments. So to realize such DNA-based cellular recording systems, we focused on the CRISPR-Cas adaptive immune system that has provided a variety of tools already to write and edit synthetic information in living cells. I'm pretty sure the audience is familiar with this system, so I will just briefly describe the whole process again. So when bacterial cells get infected by foreign DNA elements like bacteriophage here, they respond using various CRISPR-associated proteins. First, CRISPR adaptation machineries, such as Cas1 and Cas2, they take a DNA piece of the virus and insert it into a specific location of the host genome called CRISPR array. And at a later point in time, the CRISPR system can recall that memory in the form of a guide RNA that directs CRISPR interference machineries such as Cas9 to cut and destroy the virus. So while many were focusing on this CRISPR interference mechanism to develop new genome engineering applications, we've been trying to find a new opportunity from this CRISPR adaptation mechanism, because to us, it looked like a great starting point to develop a synthetic DNA-based recording system. And one thing to note here is that since Cas1 and Cas2 always inserts new spacers at the leader proximal end and push older spacers to the backwards, of the CRISPR array. The spacers in the CRISPR arrays naturally contain not only information about what invaded the cells before, but also the relative timing of the past invasions. So using this CRISPR adaptation mechanism, we developed a framework called TRACE to record user-defined signals into the genomes of a bacterial cell populations. So briefly, the system has a sensing module that converts a desired biological signal into a change in copy number of trigger DNA. And at the same time, the system has another module for writing that overexpresses Cas1 and Cas2 to constantly expand genomic CRISPR arrays with new spacers, which can be acquired either from the trigger DNA when the signal is around, or from background DNA that is also present inside of cells when the signal is absent. So by looking at the spacer content in the CRISPR array, we expected that we might be able to reconstruct the molecular history in the target environment, whether the signal was present or absent. Also, as I mentioned in the previous slide, CRISPR arrays acquire new spacers in a unidirectional manner. So space ordering the CRISPR array was expected to inform us about relative timing of each event as well. So in this slide, I will describe a little more detail about trigger DNA design in our system. So the trigger plasmid, which we call p-trig, works with two different origin of replication. So first, we put mini F origin of replication, that is for maintaining the plasmid at low copy in the absence of the target signal. And we also put another origin of replication that was derived from a phage lytic replication system and placed it downstream of an inducible regulatory element. So when the signal is around, this regulatory element was expected to drive gene expression of the phage lytic replication system to transiently increase the copy number of the plasmid. So to demonstrate this approach, we first constructed a version of p-trig with a popular lock promoter, which can sense lactose or IPTG level in the environment. And we confirmed that the plasmid copy number could be increased up to 400-fold with IPTG in a dose-dependent manner like this. And since p-trig design is pretty simple and modular, we could replace the regulatory elements with any other inducible regulatory elements. So we could develop a variety of sensors for many different signals that are relevant in the gut environment, including sugars and high-fat diet, heavy metal, oxidative stress, and also pathogen infections. So again, while the p-trig senses target signals and fluctuates its copy number in the cell, overexpressed Cas1 and Cas2 was expected to constantly expand genomic CRISPR arrays with new spacers acquired from any DNA sources in the cells. I would like to note that the chance that a certain DNA gets picked up by this Cas1 and Cas2 complex depends on the relative abundance of the DNA sequence in the cell. So the p-trig copy number fluctuations was expected to be reflected in the pattern of the new spacers in the CRISPR arrays here. And to analyze the expanding CRISPR arrays, we amplify the leader proximal end of the CRISPR arrays where new spacers are getting incorporated. And when we run a gel of the PCR products, we could confirm the mixture of unexpanded and expanded CRISPR arrays with new spacers. And then we further mapped where these new spacers were derived from using deep sequencing. And we could find that presence of target signals, such as IPTG and oxidative stresses here, could result in much more prevalent P-trig-derived spacers in the CRISPR arrays in the cell populations. We then further asked if we could encode temporal information of signal fluctuations into the CRISPR arrays over a more extended period of time. So for this proof of concept experiment, we exposed cells to different signal induction states here with or without IPTG or 1 or 0 here over 4 sequential days, constituting all 16 combinations of temporal binary profiles. And over the course of the experiments, CRISPR arrays were expanding with new spacers constantly like this. And then we sequenced the CRISPR arrays from the last time points. And then we determined the content of the trigger-derived spacers at each positions of the CRISPR arrays like this. So this is the example data. Here, L1 to L5 arrays indicate CRISPR arrays with 1 or 2 or even 5 more new spacers in the CRISPR arrays. And the left side of the plot is for leader proximal end or 5' end of the CRISPR array. And the right side of the array is for the distal end or 3' end of the CRISPR array. So first, when we compare the CRISPR arrays in cell populations that were given or not given the signals throughout the 4 days, we found that the P-trig spacers were incorporated only when the cells were exposed to signals. We then checked these 2 additional populations that were exposed to signals only at the last or earliest time points. So here you can see the P-trig spacers are present only at the leader proximal end because the cells were given the signals only at the last time points, which is more recent timing for the cells. And on the other hand, this cell population that was given the signal only at the earliest time points, it had P-trig-derived spacer only at the distal end of the array because they were incorporated first and then pushed backward by newer spacers over time. We further checked the cell population that was given the signal at both the early and last time points. And this cell population, we could detect P-trig spacers at both leader and distal end of the CRISPR arrays with bimodal pattern. And we further analyzed the array populations for all 16 combinations of temporal profiles and found the previous signal exposure profiles could result in unique pattern of the CRISPR arrays in these cell populations. And to better delineate and more quantitatively understand the data structure in CRISPR array populations, we further categorized the observed individual CRISPR arrays in the cell population by mapping the source of the spacers at each position of the CRISPR arrays. And then we quantified each of these array types in a given cell populations. So here, in addition to IPTG, we also tried recording oxidative stress over 3 sequential days like this. And we found each temporal information of oxidative stresses was embedded within the cell populations as a distinct distribution of the array type frequencies. And we further repeated this experiment multiple times and mapped the data onto this PCA space to find 8 distinct clusters that differentiated all the 3 temporal profiles from each other very well. This means the CRISPR arrays indeed have very unique patterns based on the temporal exposure profiles it recorded. And leveraging these unique patterns of the array type frequencies, we built classifiers to predict the given signal profile for the observed CRISPR array data. So here we trained a random forest classifier on the first 3 data sets, and then tested its performance on newly acquired data sets from 6 additional independent experiments. So this model produced about 94% accuracy out of 48 tested samples with TRT, array type frequency, as the leading feature for classification. And this result demonstrates that we can now call what temporal signals the cell populations were exposed to in the past by just looking at the CRISPR array populations in the later point in time after all the process is done. Then we asked if these data contained in cells are stable or not. So for this, we propagated the data containing cell populations over 100 generations and tried to reconstruct the data at multiple time points. And we found that we can read the data even around 80th generation with about 90% of accuracy. And we also challenged the data containing cells in soil environments where many DNA degraded factors exist. And we also tested naked DNA that has the same amount of information as a control here. So while the naked DNA lost all the information during the 6-day incubation period in soil, data contained in cell could be protected by the cellular envelope and did not lose any information for the same period of time in the open environments. But there is still a long way to go for this technique to be practical and useful to measure information in native microbiome environments. The first problem we have is that the lab E. coli strain that we have been using for all the previous application is not good host for in vivo applications. So even when a ton of cells, about 10 cells, were orally gavaged, they cannot colonize the gut at all, and we cannot detect them within a day or so. So we can think about porting this system into better gut colonizer strains like E. coli nasal or human or mouse isolated commensal strains. But when the recording system is introduced into those strains, its efficiency or activity decreases more than 90%. And this level of activity is not enough for efficient recording of biological signals. So for example, the gray data here in this plot is showing distribution of arrays with different number of new spacers after 3 days of recording in vitro. And as you can see, a very limited proportion of cells have arrays with multiple spacers, which contain most temporal information. So we usually try to enrich these rare long arrays by bead-based size enrichment, but this is not useful when the population is already bottlenecked. So we wanted to improve the utility and data storage capacity of our system and engineered Cas1 and Cas2 proteins for higher activity. So for this, we devised a directed evolution strategy for CRISPR adaptation processes. So first, we generated a mutant library of Cas1 and Cas2 by error-prone PCR, and then introduced the library into the E. coli strain with this synthetic reporter system in their genome. So in this system, I placed GFP and kanamycin-resistant gene upstream of CRISPR array in the genome. And then I electroporated a synthetic oligonucleotide with T7 promoter sequence and expected if some Cas1 and Cas2 variants in the library have higher activity than others, then they will likely incorporate this T7 promoter oligo as a new spacer into the CRISPR array, and then they will survive the kanamycin selection step in the pipeline. And long story short, we could isolate 2 variants from the screening, and they showed significantly higher activity than wild type Cas1 and Cas2 complex, about 5 to 6-fold. And then we examined if the variants could capture biological information in higher temporal resolution. So for this, we exposed the cells to various durations of paraquat-induced oxidative stress pulses. And the oxidative stress was sensed by cells using p-trig under control of SOX-RS, redox responsive regula. While both wild type and evolved variants could resolve oxidative stress pulses with longer than 3 hours pretty well, oxidative stresses induced by shorter pulses could only be captured and resolved by highly active variants only, highlighting higher temporal resolution of the evolved variant for recording biological events. We also examined if the variants could capture biological information with higher sensitivity. Here, we exposed the cells to various levels of paraquat-induced oxidative stress pulses. And similarly, while oxidative stresses induced by 6 micromolar or more of paraquat could be resolved by both wild type and evolved variants, weaker signals could only be captured and resolved by the variant with much higher sensitivity. And beyond the higher temporal resolution and sensitivity, we found that the evolved variants are more functional in many different bacterial strains. So in all these tested 6 strains, the evolved variants show much higher activity than the wild type proteins. And what is exciting is that now we see highly active CRISPR array expansion in these EC257 and EF strain, which are the isolated strains from the gut environment of human and mouse. So we are expecting they will perform better with higher stability in the mammalian gut environment. Also, this Klebsiella oxytoca strain is a soil microbe, suggesting there are applications for biological signals in the soil, such as plant microbe interactions. I'm very excited by the potential application of our recording system in vivo. Currently, we are working on recording these 3 different types of biological data in the mammalian gut. So first, we are expanding sensing capability of our trace system by mining and testing transcription factors from bacterial genomes. And second, I'm using CRISPR array as continuously evolving barcodes of bacterial cells to track their lineage information during bacterial adaptation or pathogenesis in the gut. Third, I'm using CRISPR spacer acquisition to capture prevalent but transient events of horizontal gene transfer in the mammalian gut microbiome. So all these are currently ongoing projects, and due to the limited time for this talk, I would like to show you some brief teaser for new lineage tracing application of CRISPR-based recording. So mice show very interesting behavior that they eat their own feces to enable them to absorb essential nutrients such as vitamins. And gut bacterial population in a mouse can be transmitted very easily to another mouse that share a cage through this behavior. So I wondered if we could track this bacterial transmission between co-housed mice over multiple generations using continuously expanding CRISPR arrays with unique new spacers. So for this, I introduced the recording bacterial population to a single mouse first, and then this mouse was housed alone for 3 days, then co-housed with 2 other mice for another 3 days, and then each of these mice were co-housed with other mice again over the course of 3 rounds of co-housing, resulting in a defined branching history of gut bacterial population across these mice. Then I collected fecal matter from all mice and sequenced the CRISPR array populations from the samples. So, and here I use simple Jacquard metric to assess similarity of the new spacers between the different CRISPR array populations across the mice. And interestingly, the CRISPR array population could be used to reconstruct a co-housing history of mice accurately for both generation 3 and generation 4. And this shows continuously expanding CRISPR arrays can contain not only information about targeted environmental signals, but also lineage information of the bacterial cell populations, suggesting new application area of our system. Then we further explored whether we could track the evolutionary dynamics of bacterial cell population within a mammalian gut. So for this purpose, we tracked gut bacterial population using a recording system for approximately 100 days. And we measured the number of bacterial cells in the gut, and their CFU levels remained almost constant throughout these 100 days experiments. This constancy could suggest that these populations are quite stable and coexist very peacefully. But when we sequenced their CRISPR arrays and analyzed the new spacers, we observed very interesting dynamics within the cell population in the mammalian gut. So in this plot, each CRISPR array is color-coded based on their new spacers at the leader proximal end. And the emergence of new colors like this within the same hue indicates that they are derived from the same ancestor. So data revealed that there was intense competition within the seemingly stable bacterial populations, leading to rapid domination by only a few successful lineages here. And excited by this novel type of high-resolution lineage tracing data, I'm currently conducting further analysis on these barcoded cells to track the mutations they accumulated step by step during the adaptation processes in the gut. And I expect that these continuously expanding CRISPR arrays might offer a powerful strategy to track bacterial populations over the course of evolution, potentially opening new avenues for understanding molecular behavior and developing targeted treatments. And beyond my current efforts, I believe a DNA-based cellular recording system can be deployed in a variety of useful ways in both basic research and also applied settings. So we can imagine this kind of DNA-based recording system can be ported into multicellular organisms also to study how they develop and how they signal each other. Or maybe this could be used in applied settings to monitor our health or our surrounding environments as well. And there have been a number of people who have participated, mentored, and supported this work, especially my postdoc advisor, Harris Wang at Columbia University has been a phenomenal supporter of my personal and also academic growth. And I would also like to acknowledge all the Wang Lab members for their input throughout the projects, especially Ravi, Yiming, Ross, Jasmine, Deirdre, and Andrew. And I would like to thank my lab members as well here. With that, I'd like to conclude. Thank you very much for your attention. I would be very happy to take any questions.
Fantastic, fantastic talk. It's amazing. You made a lot of progress, even I guess you're doing within 1 year in your lab as well. Continuing amazing work you started with the Harris one.
Thank you so much.
Thank you. Yeah. I want to say, Dave, do you have any question, comment?
Yeah, I actually have a technical question. I mean, the DNA recording is so cool. I love the ability to say track the microbes between mice. Do you want to can you give us an update on RNA recording and the possibility of engineering that and being able to track expression states of the microbe too?
Oh, right. So RNA recording system. So I guess some research labs are focusing on recording like RNA expression levels at like transcriptome scale using reverse transcriptase associated CRISPR-Cas1 and Cas2 systems, and it's a really fascinating system. I guess Randall Platt group is working on this system, and we are really excited about it. But the system is recording any like unstructured RNA inside of cells, so it's not very focused. So to get all the information out of the cell population, they may need a lot of sequencing reads.
And so the challenge is going to be detecting the right signal among this huge background.
Right. So our system is actually focused for like 1 or like 3 or 4 different signals here to guide a targeted signal into a change of triggered DNA copy number here. So I guess there's different applications for 2 different systems we have here.
Great.
Okay.
Well, very impressive work. Yeah, look forward to seeing all the different creative uses of it.
Thank you so
Dave,
much.
Do you have any other question or comment, Dave?
No, that's it. Great talk. Yeah, it was
Okay.
really fascinating to see.
Thank you. So let me ask some question whether that would be possible or probably challenging. So I know, your group also working on, the Harris One group working on multiple different projects. Actually, I attended microbiome conference, the last weekend, and then there are many Columbia University folks gave a talk, also, present posters, and then one idea I just got, during your talk was, they use the conjugation-based magic system or improved version of magic system, including, I don't remember the name, but KAIST something. And then you have the recording system. And then by combining these 2 things together, you may have something amazing, kind of transfer the recorder to whatever member of the community and do something amazing. Have you thought of those things, or anyone working on this type of thing? In the Harris group or some other group?
Right. That would be really interesting to port the system into native microbial community and make them record something very interesting. But the limitation, so we've been brainstorming about this idea for a long time.
Oh, you did. You guys did already.
Right. But the current issues here is that since this Cas1 and Cas2 based CRISPR recording system relies on some of host factors in this E. coli strain, so so we've been trying to port the system into like other bacterial species, but we've been only getting like successful results from very closely related species because because this particular CRISPR array requires E. coli's integration host vectors, or IHF,
Mm-hmm.
for the targeted integration of new spacers by KAIST1 and KAIST2.
I see.
So I guess that's the hurdle here. But
Mhm.
once we get some idea of how to port all the system together with host vectors into
Mhm.
other species, that might
Mhm.
be very exciting.
Yeah, the reason I'm asked that question is I remember you show one slide, the many different, strain or bacteria, the recording system sort of work in some sense, right?
Right.
Is it all of that is the E. coli or some of this not E. coli? I don't remember that.
So
Oh, some of
here,
them, yeah.
yes. So 3 of these strains were E. coli strain.
Yeah, you have the other thing.
And this is a Fergusoni strain, which is pretty close to E. coli.
Uh-huh.
And this is Klebsiella. 2 of these are Klebsiella strains.
I
So
see.
they are like very, very close relatives.
Related to the E. coli, so but
Right.
at the end of the day, you could expand that one to potentially other bacteria, hopefully.
Right. So
Yeah.
we've been successful porting these into Pseudomonas and Vibrio
Wonderful.
and Pentoia, but not like in gram-positive yet. So
Yeah.
yeah.
Oh, that's a completely different story, so more challenging, I guess.
Right.
Okay, absolutely amazing. I really looking forward to seeing much more great paper from your lab in the future. Thank you.
Oh, thank you so much.
So let me check whether we have question from audience. But again, this is the final week, so I don't see many people today. Okay, so let's close today. So thank you all for joining and staying today. We'll meet again after a long break. We'll meet on January 11, 2024, Thursday, the same time, so same Zoom link. We'll have Professor Hadley Sykes at MIT and Dr. Luan Liu at Nanyang Technological University. Oh, another 1 a.m. or midnight person. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote YouTube panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. And also, happy holidays to you all and New Year as well. Thank you.